Throughout the lessons on the ovarian cycle and the menstrual cycle, we've spoken a lot about if fertilisation does occur and if fertilisation doesn't occur. But in order to lead us down the future pathway of human reproduction and looking at the development of embryos, we need to now understand what happens if fertilisation does occur. So in order for fertilisation to occur, sperm must be, I don't know what's happened there with the highlighter, but in order for fertilisation to occur, sperm need to be brought into contact with the ovum. And in order for that to take place, sperm needed to be deposited into the vagina and the male must develop an erection. So we've previously spoken when we spoke about the male reproductive system about what an erection is, but very simply it's where that spongy muscular tissue within the penis becomes engorged on blood and it allows it to penetrate into the vagina for sexual intercourse. So if we look at the pathway of sperm here, sperm start down the bottom here. I'm going to use a light blue pen, should be easy to see. Start here in the testes. They then go into the epididymis to mature before they're passed out by the vas deferens, go over the top of the bladder, and these sperm are then met by the release of sugary fluid from the seminal vesicle, and then they pass through the prostate gland, which release alkaline fluid, and they're also met by the bulbourethral gland, which helps to release some more components of our sperm or semen, before this continues to flow out via the urethra now. And as we can see, the penis is in the vagina here. Sperm is deposited at the cervix, where I'm drawing my large blue dot now. Sperm will then flow up through the urethra, and then they will travel up. In this diagram, it only shows one, but they will travel up the fallopian tube, or the uterine tube, or the oviduct, and this is where fertilisation is going to occur. So in the uterine tube itself, this is the site of fertilisation. It's what we're going to have a look at in detail today. So to talk about fertilisation, we first need to very quickly talk about this ejaculation and this sperm being passed out through the penis itself. And so once the penis has become sufficiently stimulated within the vagina, the epididymis, the vas deferens, the seminal vesicle, and the prostate gland are all going to contract, and that's going to lead to sperm or semen being propelled out of the body. That process is known as ejaculation. And in ejaculation, normally about 3 mils of semen is deposited. Now, that deposition of sperm into the vagina is actually known as insemination. So ejaculation is where semen is propelled out of the body. Insemination is where sperm are deposited into the vagina. So when we talk about the components of semen, we briefly mentioned them in our diagram, but in terms of sperm, they're produced by the testes, they mature in the epididymis, their job to fertilise the egg. In terms of the seminal vesicles, they release that sugary fluid, makes up about 60% of the overall fluid of semen, and its job is to nourish and provide energy for the sperm themselves. In terms of the prostate gland, we mentioned that it releases that alkaline fluid. Now that alkaline fluid is to neutralise the acidity of the vagina. And lastly, we briefly alluded to it, but the bulbourethral gland is there to help to lubricate the head of the penis for sexual intercourse. So when we're having a look here at sperm's journey, we've gone through this in the diagram. The only thing that I want to draw attention to here is that only a few thousand sperm actually reach the fallopian tubes. There is a very, very high chance that most of the sperm are going to die due to a number of factors when leaving the body. So think that high acidity within the vagina, think them not finding the egg, they've also got a very short lifespan, they might not have enough energy. There's 101 ways that sperm won't actually make that journey. And as a consequence, it's really important there are lots and lots of sperm. So, in order for sperm to fertilise the egg, we need to understand what is surrounding that egg, what is surrounding that secondary oocyte. So in our lesson on oogenesis, we spoke about the secondary oocyte and we spoke about this particular cell, this gamete, freezing during the second meiotic division at metaphase. And at this stage, the oocyte itself is actually surrounded by 2 really important layers. The first layer, which I'll highlight in green, is known as the corona radiata. And the corona radiata are all of these smaller little follicular cells which surround that secondary oocyte. Now, these follicular cells are held together by something that we're going to call a cementing material, and this cementing material contains an acid to help protect the oocyte itself. The second key layer that we're going to have a look at, and I'll highlight this one in blue, is what is known as the zona pellucida. And this is that inner layer, I accidentally unhighlighted there, but this inner layer that I'm highlighting in blue was originally in yellow. This is known as the zona pellucida. And that is that inner membrane, it is known as a glycoprotein matrix, so like a sugary protein matrix, and that surrounds that cell membrane of the oocyte. Now, when we talk about these two structures, they're really important to not only protect the oocyte, but now the sperm have to break in past these layers in order to fertilize this egg. So in terms of fertilization, let's talk about how this happens. I'll get rid of some of this leftover writing. Now, step one, the sperm arrive at the ovum. So I'll try and highlight each stage in a different colour here. So firstly, sperm arrive at the ovum. That's probably the toughest bit for a lot of the sperm, is actually finding the ovum. Number two, in green, is that the enzymes in the head of the sperm, enzymes in the acrosome layer, begin to break down that first outer layer, that corona radiata. So I'd previously highlighted the corona radiata in green, as we can see up in this diagram here. This is what the sperm has to get through first. Now, one sperm is not going to do the job. In fact, several thousand sperm are needed in order to provide enough enzyme to break down that corona radiata. When sperm make their way through the corona radiata, they now encounter that zona pellucida. So we can see in step 3 here, this is the sperm cell, and it is encountering that zona pellucida. Now, enzymes in the acrosome, again, help to break down the zona pellucida, and it gives sperm access to that plasma membrane. Now, the plasma membrane we haven't specifically spoken about, but it's just that outer lining of the cell, or of the egg cell itself, and it is where that oocyte and the spermatozoa fuse. And it's during this time that when that sperm enters the ovum, it stimulates the formation of what we know as a fertilization membrane. And this fertilization membrane prevents other sperm from fertilizing this egg. Once these sperm and egg have fused, it is then known as a zygote. Now, you'll see up in this diagram I've highlighted egg and sperm nucleus. What these can also be known as is the egg nucleus may be known as the female pronucleus or pronuclei, and in the instance of the sperm nucleus, that will be known as the male pronucleus or male pronuclei. So that there shows and depicts the process of fertilization and the formation of a zygote. Now, before we leave this lesson, we need to make sure that we understand 3 really important terms. That first term that we just spoke about then, I'll use the same colour, is zygote. And a zygote, very simply, is a single diploid cell. It contains DNA from both the mother and the father, and it forms immediately after that fusion of the male and female pronuclei. Okay, so immediately after fertilization. Let's just tidy this up a little bit here. So zygote, diploid cell, mother and father. So this here is our zygote, down the bottom in that image. Next, from the zygote stage, it's then going to continue, or this cell is going to continue to divide thanks to mitosis. And in terms of mitosis, it's going to continue to divide up until the 8th week of pregnancy, and it's during this time that we know this particular set of cells as an embryo. After 8 weeks and until the end of the pregnancy period, it will then be known as a fetus. Okay, and this fetus is going to look somewhat like this in the early stages, and then will continue to develop into something that looks more and more like a human baby.